Good afternoon. My name is Chris Griffiths from the University of Manchester, and I'm delighted to welcome you to this edition of the ESDR Kitchen. Those of you who have been following us will know that these have been going now since July of last year, when the ESDR board made the decision that we needed to stay in contact with our membership from both an educational and scientific point of view. And we launched these webinars, which are still going strong every couple of weeks. As you can see here, we have 4 tracks. The recipe book, which is where someone discusses new research techniques explained or made simple, as we often read in the JID. Sweet and Sours, where we bring in 2 experts to discuss hot topics in dermatology research, sort of a spirit of scientific discourse. Freshly Baked is literally as it says. These are recently published high-profile papers, and we asked the first authors of those papers to discuss with us the key outcomes and what these papers mean. And the 4th is Molecular Cuisine, which is what today's theme will be about. And these where a research leader in dermatology and skin biology will take us through his or her journey in discovery. If we go to the next slide. So today is Molecular Cuisine, as I've said, with Lars who will be introduced by Sprecher. And this today's session is sponsored by Elmrel and supported by Bristol Myers Squibb. Over to your chairman for today, Sprecher.
Thank you, Chris. Hello, everybody. So it's my great pleasure to introduce our speaker of today, Professor Lars French, who actually doesn't need any introduction. So we'll make it relatively short, much shorter than what actually could be said. So Lars is chairman and professor of the worldwide renowned Department of Dermatology and Allergology at the Ludwig Maximilians University Hospital in Munich. He's one of the most remarkable role model as a clinician scientist. He graduated from the University of Geneva, trained in internal medicine, basic research, and dermatology at the same university. Professor French and his team are engaged over the years in research and development in the field of inflammatory skin diseases, adverse cutaneous drug reactions, and tumor immunology. Was the one who discovered the role of fast ligand in the pathogenesis of TEN. He has co-authored over 320 original publications. He has an H index of 54. He's ranked by Stanford University among the top 2% scientists globally. In 2012, Professor French was awarded the Autoinflammatory Prize for the promotion of medical research, one of Switzerland's most prestigious scientific awards. He is past president of our society, of the European Society of Dermatological Research and of the European Dermatology Forum. He is a member of the German Academy of Science, and he's currently the president of the International League of Dermatological Societies, or ILDS. I will pass the podium to Lars in a minute. I just want to remind you to forward all your questions to the chat or the Q&A function. Lars, thank you so much for your willingness to contribute another meal to our kitchen. Please, the floor is yours.
Thank you very much, Ellie. Thank you, Chris, for the wonderful introduction. Thank you to the board of the ESDR for giving me the occasion to participate in this innovative series, the ESDR Kitchen and the Molecular Cuisine. I'm happy to be again together with you with the ESDR. We really miss the ESDR meetings live, and I'm longing for a live Congress again now that the COVID situation is slowly getting better around the world. I will here talk to you a little bit about my voyage or my personal expedition in research as a clinician scientist. And obviously going through this, I won't be able to go into great detail scientifically for each paper, but more try and illustrate the path that I have taken. It may be instrumental and interesting to some, but as wise people say, there are many roads that take you to Rome. This one may be one that gets part of the way to Rome, but there are certainly other ways of doing it. So my disclosures first. These are my current disclosures. And here is the start. In fact, I had a very atypical start. I studied medicine in the University of Geneva. I did the international school in Geneva and English speaking, and then went into medicine speaking French. And in the fourth year of medicine, you get courses from different professors in the subspecialties, so not just surgery and medicine. And one of them that was incredible from a didactic perspective was dermatology. We had 1 hour a week, and I remember I used to always fight to sit right at the front because it was so fascinating. And that was Jean-Hilaire Saurat, who many of you know, who gave the course for students and made a huge effort to teach dermatology in an interesting way already for students, linking the challenges that are still open, the science that was developing, and not only the clinical side, but also the pathogenesis of disease. I rapidly decided then to change one of my rotations that I could have in the 6th year of medicine, and I swapped in, I think, gynecology for going into dermatology, but I couldn't get clinical dermatology. The only rotation I could get as a student still was research. And I decided really to take 4 months and go into research in dermatology. And there, one of the senior physicians and dermatopathologists working in Geneva with Professor Sora was Yves Méraud, and he was my first mentor and gave me a project. At the time, there was a paper that had come out that suggested by looking at CD4 and CD8 staining, at the time it was called OKT4 and OKT8, you could distinguish lupus from Yesner-Kanov's lymphocytic infiltration of the skin that you can see here. And he said, Lars, we have the cases. Can you go and do stainings and see if you can reproduce what's being published in the literature? And yeah, I worked very hard for the 4 months, had a lot of fun, found it really great. And I was able then to show that that is not the case. You can't differentiate. And then Yves Méraud wrote the letter. I think that was the first lesson for me, is don't believe anything that's in the literature, at least if you're going to set out on a big project or a grant. Do preliminary work to see if you can reproduce key elements of what is in the literature, or look if it's being reproduced by others. That's really important. So from there on, I finished my medicine, and then I decided to go into internal medicine. And Professor Sora had already said, you can have a residency in my department, just tell me when you want to start. And I told him I'm going to do internal medicine. He said, you'll see, that's nice for 3 months, but afterwards it'll start get boring. Dermatology is much more interesting. But I loved it. The 2 years I was there, I had 2 mentors, Professor Müller, who was a very strong internal medicine professor and nephrologist, and Professor Schifferle, who was a clinician scientist in internal medicine and nephrology. And I did several rotations, a lot of ward, and enjoyed that a lot. Thought about staying and was offered a position to stay. I really liked being in emergency care, but Professor Sora kept on saying, you know, emergency care for a lifetime, that's not really something that's fun. And down the line, I did want to do research and decided to go down a path of doing more research. And that's where I joined, in fact, the lab of Jörg Schifferle, who was a very good researcher working on complement and kidney disease. And he interviewed me and took me into his lab and gave me quite some freedom to work. The first project he gave me was on a newly identified protein that Professor Jörg Chopp in Lausanne had discovered. And Jörg Schifferle had met him on a meeting, and this comes to a second important fact. Networking and meeting in meetings with people is really essential for developing good research projects. And Jörg Schifferle came to me and said, listen, I just got a really original cDNA and antibodies against a newly identified protein called clusterin, which is supposed to be an inhibitor of the terminal complement phase, the membrane attack complex. your chop would be interested in knowing the tissue distribution. And so I set out to do staining experiments. First of all, we did quite some work in glomerulonephritis and could show it's upregulated in glomerulonephritis and co-localized with C9 of complement and C3. But then collaborating with a lab next door who's doing a lot of in situ hybridization, the lab of Professor Sapino in the oncology department, I decided to use that cDNA and learn how to do in situ hybridization. At the time, a technology that was done with tritium labeled probes, as you can see up at the top, this like star bannered sky in panel B, that was the RNA, mRNA for clustering in the thymus, in the human thymus. And we had to incubate these slides with tritium for 2 to 3 months. So an experiment took that much time, and you had to decide when you take the slides out at the right moment to get a good signaling. Now the technologies enable you to do that in 1 to 2 days with other staining procedures. But this was really interesting, and by studying this molecule, we realized that beyond having a role in complement, it may well have a role in programmed cell death. And I was able to show that clusterin was confined to surviving cells during the process of programmed cell death in the thymus, but also in models of programmed cell death, and also during embryogenesis. You see these beautiful images of whole mounts of mice during embryogenesis showing where the gene is expressed for clusterin most strongly. And these were regions where there's a lot of remodeling, and during embryogenesis, programmed cell death is essential. For example, for us to have spaces in between our fingers, the cells during embryogenesis between the fingers have to die by programmed cell death. This got me really interested into the field of programmed cell death, which was exploding, also called apoptosis now, exploding at the time. And having been quite successful during my 3.5 years in Professor Schifferle's lab, I immediately before leaving his lab wrote my first research grant and was able to get a research grant from the Swiss National Science Foundation and joined the Department of Dermatology with my own grant and was able to build up my own group. And this is a third message that's really important. I'll say it again at the end. That choice was really a really good choice at that time. If you're going into research as a clinician who wants to do research, you need support. And there's many good centers around the world. There's a lot of good chairmen around the world that I know who do provide this support and do provide you protected time, do help you if you have hard times, do help you learn your clinical talents more rapidly by showing you interesting, important cases. And this was really the case in dermatology with Professor Sora. So I could set up my lab and do my residency training in parallel. And I remember he would come and get me when I was doing in the afternoon lab work. He would pick me up and then take me into the clinic and say, hey, look at this. This is poikiloderma, and explain it to me. And then he'd let me go back to the lab. So yeah, really a fantastic mentor from this perspective. I then really started getting interested in trying to understand in which skin diseases could apoptosis have a role. And that's where I started working on adverse drug reactions and toxic epidermal necrolysis. As you know, apoptosis is important in the early stages of epidermal detachment in the adverse cutaneous drug reaction, toxic epidermal necrolysis. But it is not known how this apoptosis was induced. We knew that often in the histology, you don't find many cytotoxic lymphocytes. So the question was it these lymphocytes being very active and running around the epidermis and killing keratinocytes massively, or did some soluble molecule or membrane-bound suicide molecule play a role here? And we looked at this in more detail. At the time, and this was in 1993, what we knew about apoptosis was a lot less than now, but the death receptor family, members of the TNF family had been identified, FAS and TRAIL. It was known that growth factors and loss of cell extracellular matrix interactions could trigger cell death, that metabolic and cell cycle perturbations could do that too, as well as DNA damage. and that the process was executed actively by caspases. And here again, collaboration was also important. I continued through my work on clustering. I developed a really good friendship and collaboration with Professor Jörg Schopp, who was one of the leaders in the field of death receptor mediated apoptosis. He cloned several of these molecules, and I would meet with him once a week in Lausanne, and we would discuss projects. He would tell me about data before it was published, and I would take it back and try and think how I could apply that to skin research. And I had a lot of reagents available. And rapidly, I looked at different death receptors and ligands, their expression in the skin in toxic epidermal necrolysis, and was able to find a strong level of expression of the protein coding for the cytolytic ligand, fast ligand in the skin of TN patients, as you can see here, and conserved expression of FAS. So everything was there, all ingredients were there in principle for skin, for keratinocytes via FAS ligand to kill adjacent keratinocytes, signaling through the death receptor FAS. And we set out to look at this in vitro. There's no good mouse models, but we would take skin sections and we would put, so these were skin sections from toxic epidermal necrolysis patients or controls or maculopapular rash patients, and incubate target cells on these for a few hours and see if apoptosis would be induced. And as you can see in this graph on the left side, there was quite strong apoptosis induced by 10 skin sections, T and C skin sections. And this could be blocked by co-incubating with a monoclonal antibody that blocks FAS ligand, showing that it was FAS ligand that was inducing apoptosis in this situation. And we put up then this model of the pathogenesis, where for reasons that we did not understand at the time, keratinocytes who do have FAS ligand intracellularly upregulate FAS ligand expression and express it at the surface of keratinocytes and thus kill adjacent keratinocytes by death receptor signaling. In the meantime, it's been shown that apoptosis really is important. Necroptosis is also important in lyell, and that other molecules alongside FAS ligand do play an important role, such as granulysin and also annexin, for example. From there, then comes to a situation where my mentor was also, being a great clinician scientist, was instrumental. And also this highlights the effect of serendipity and opportunity in science. We had a challenging clinical case, and Professor Sora, it was as usual on a weekend, was called by a senior physician. This patient here developed toxic epidermal necrolysis and was treated with high dose 1 gram a day methylprednisolone for 5 days. And as you can see, he got worse under treatment. And Professor Sora was asked by the intensive care doctors, is there anything else we could do? And he suggested, well, the only thing I could suggest is that you try IVIG. And within a day, this patient changed and he ended up surviving. And we visited this patient together. And based on this, he told me, listen, you should look and see if IVIG does anything in your model of apoptosis. And this is what we did. We went back to the lab and I had a wonderful postdoc here, Isabelle Viard, who looked at this, and she did the following experiment. She took different types of cells in culture, primary keratinocytes, HEK, or cell lines on the right, exposed them to recombinant soluble FAS ligand, and pre-incubated the cells either with albumin or with IVIG. And when you pre-incubated cells with IVIG, you could completely protect them from the effect of FAS ligand, recombinant FAS ligand. And we could then go on and show that intravenous immunoglobulin do contain quite high titers of neutralizing anti-FAS antibodies in them. And this explains, in fact, the inhibitory function. We went on in Switzerland to treat 10 patients with IVIG. It's a very small series, doesn't really give you a strong conclusion, but with a mortality of 20 to 30% of the time, we were encouraged to see that none of these patients did die, all survived under IVIG therapy. And we were able then to publish this in Science at the time. And since then, we've gone on, we've been able to show that it's an upregulation of inducible nitric oxysynthetase that leads to fast ligand upregulation. And we were also able then to do other clinical trials. It was not possible to do controlled trials, but these were trials where several centers accumulated their cases. And the situation now is that there is no consensus if IVIG are really beneficial or not. There's, I would say, 50% of the trials that do say there's an effect. There are some nice meta-analysis that show that it's dose dependent. If you treat patients with over 2 grams per kilo, they do have a survival benefit. But if you take all studies that are in the literature as done here, the only therapy that does seem to be pan out with a benefit is corticosteroids with IVIG together. The problem with IVIG is that some of the trials are using very low doses. Some of the patients are getting the treatment when they're completely detached already and there's no cellular apoptosis to block. So you would need to study this in a more controlled manner or with much larger series of patients. And so going on from that, although I left this field of research somewhat, as you will see later, I did go on with to set up an international registry for toxic epidermal necrolysis, which you can find online, together with colleagues in Miami, in Kyoto, and in Niigata University, and in Paris. And we are now recruiting patients here. Any center that sees these patients is welcome to apply for collaboration. You will be acknowledged on the website and can have access to the data. There is artificial intelligence in the background computing the data, and there's optional biobanking associated with the registry. And with this, we hope to have a larger series of 100 cases per year coming into the registry, and down the line be able to say, is there anything that is better than supportive care in a pretty unbiased manner for a disease that's extremely rare? This occurs in 1 patient per million per year. Yeah, after doing this work, I moved to the US for 1 year, a fantastic year, as a fellowship in the department of Professor John Stanley at the University of Pennsylvania. And I worked together with another mentor who was fantastic, Professor Alan Rook, in the field of cutaneous lymphomas, and interested myself with clonality and the effect of clonality of T cells in systemic sclerosis and in the response of these diseases to photopheresis. Here you see in the middle of this group, Alan Rook, and you see we would do rounds in the cutaneous lymphoma clinic, and he was one of the biggest managers of Sézary syndrome patients. I worked also on Sézary syndrome and could show that impaired CD40 ligand signaling is a cause of defective IL-12 and TNF production in Sézary syndrome. And you know that Sézary syndrome is a Th2 skewed disease, and so this may explain partly the immune suppression that these patients have. I came back then to Geneva and got a professorship there, an endowed professorship, and then moved my field of research. Jörg Chopp had discovered a new family of proteins that were similar somewhat in structure to the death receptors. And these were the NLRPs, which are members of the inflammasome. They are sensors of pathogen-associated molecular patterns and danger-associated molecular patterns, and really important cytoplasmic proteins for sensing danger, for example, uric acid crystals, but other forms of danger, and triggering the maturation of pro-interleukin 1 beta to interleukin 1 beta, or pro-IL-18 to interleukin 18. And again, I had access very early to technology and a mentor with which I could interact in Jörg Chopp, which was fantastic. And at the same time, evidence was coming out that IL-1 and the inflammasome could play a role in autoinflammatory diseases. And we quickly realized some of these diseases did have skin signs. For example, Papa syndrome with pyoderma gangrenosum and acne. And several of these diseases were characterized also by neutrophilic infiltration, as you can see on the right side in deficiency of IL-1 receptor antagonist. And so besides the neutrophils, interleukin 1 plays a very important role in these disorders of innate immunity. And we decided to look at this a little closer. I first looked to see if inflammasome components are expressed in keratinocytes, and we could find that the NLRPs 1 and 3, as well as caspase 1 and 5, and ASK, a key molecule necessary for binding the NLRPs to the caspase platform, which is needed as an enzymatic platform to activate interleukin-1, was present in keratinocytes. And then we could show that in fact, activation of the inflammasome is essential for inducing the reduce or enabling the secretion of mature IL-1 beta upon exposition of keratinocytes to contact sensitizers. And we could later show that in fact the early phase of elicitation of contact hypersensitivity to a contact sensitizer, TNCB, is dependent on the inflammasome components ASK and NLRP3 in mouse models. This was work done together with Olivier Guide and Hideki Watanabe, working with me at that time in the lab in Geneva. So I told you about this PAPA syndrome, and the PAPA syndrome is characterized by very severe acne in adolescents. The presence of pyoderma gangrenosum and pyrogenic arthritis is known to be due to mutations that influence inflammasome function and lead to increased secretion of IL-1 beta. So I set out to ask if acne could also be due to increased production or aberrant regulation of IL-1 production in the skin. And we were able to show, in fact, that Propionibacterium acnes in vitro, but also in vivo, is a strong activator of the inflammasome in macrophages. And probably when it gets released from the follicle that expands due to the accumulation of sebum in the follicle, you have some release of P. acnes, and this is enough to activate the macrophages that are there and induce strong secretion of IL-1 beta. And as you can see here, you can test that with an ear swelling model. On the left, you inject P. acnes into the ear. In the middle panel, you can see histologically an accumulation of neutrophils. And this is completely blocked if you do it in mouse that are deficient in inflammasome components such as ASK or NLRP3. And we can later show by transfer experiments, a bone marrow transfer experiment, that it's really the myeloid cells that sense P. acnes in vivo and are important in the production of IL-1 beta in acne. So from there on, I moved to looking at two other drug reactions and interesting myself at other members of the IL-1 family, and this was IL-36. And these two diseases are acute generalized xanthomatous pustulosis, a drug reaction that occurs 1 to 3 days after intake of a drug, is characterized by disseminated pustules. So again, neutrophils here present on the skin, associated with fever and inflammation. And then the very frequent EGFR MEK inhibitor acneiform eruption that occurs in near to 80% of patients taking these drugs for cancer. And unfortunately, these patients often stop the drug and their cancer progresses under this situation. And for decades, the pathogenesis was not understood. So we looked at this a little closer and using more modern techniques, so gene expression arrays for looking at the transcriptome and biopsies of patients suffering from acute generalized xanthomatous pustulosis. And you see by hierarchical clustering here how well this technology can segregate different types of diseases. You see normal skin, in the middle maculopapular rash, and then AGEP. They all cluster together with the same kind of signature. And then when you look in more detail, you see that AGEP is different to the others in that you have much stronger IL-36 and IL-8 expression. IL-8 is very important for neutrophil recruitment. We could reproduce this in qPCR in the lesions, significantly increased IL-36 gamma in the skin of these patients, and then could show in vitro that macrophages of patients, and even if you take these monocytes macrophages 1 year after the skin rash, they would, in 1 to 3 hours of exposure to the culprit drug, they would secrete massively IL-36. And this was not the case with the control drug. And we could show this with keratinocytes too. So IL-36 really does play a key role in the neutrophilic infiltration of the skin that occurs during acute generalized xanthomatous pustulosis. This is beautiful work that was done by a brilliant MD PhD student in my lab in Zurich, Dr. Weiber Meyer Schieser, a few years ago. As you know, IL-36 is a member of the IL-1 superfamily. It binds to the IL-36 receptor and signals via MYD88 to induce MAP kinase and NF-kappa B signaling with downstream production of pro-inflammatory innate cytokines that are listed here, and that mice that transgenically overexpress IL-36 have a psoriasis-like phenotype. In the meantime, you also do know that patients who have mutations in the brakes on this system, the IL-36 receptor antagonist, can develop a generalized pustular psoriasis, again, a neutrophilic autoinflammatory disease. So together with another brilliant postdoc that came to join me from Japan, Takashi Sato, we decided to look at EGF receptor and MEK-induced acne in cancer patients. And again, using transcriptomics, we could find very high induction of IL-36 gamma in the skin of these patients. You see here this beautiful immunohistochemistry showing the expression strongly upregulated in the pilosebaceous unit of IL-36 gamma. You could also show that by in situ hybridization. And in a very long set of experiments, Taka could show that you need two signals to get IL-36 upregulation in these patients. You need a commensal bacteria signal, that is Propionibacterium acnes, for example, inducing NF-kappa B upregulation and binding of P65 to a P65 site on the promoter of IL-36. And then you need another transcription factor that Taka identified by mining, and this is KLF4. KLF4 is upregulated when you block EGF or MEK signaling and binds to a KLF4 site that is right next to the P65 site on the IL-36 promoter. And by mutation analysis, he could show that both these sites are essential for upregulation of IL-36 production. And this is the cause of the pustular acneiform reaction that occurs in patients receiving MEK and EGF inhibitors. So KLF4 and NF-kappa B signaling upregulating IL-36. We published that last year in the Journal of Clinical Investigation. So some of these take-home messages I've already told you, but I'll go through them again. Being a clinician scientist is additional work and challenging, but it's a wonderful activity, very enriching with enormous opportunities. And the fact to try and understand disease better is really wonderful as a clinician. Good ideas, creativity, perseverance, and modern technology enable us to innovate. Innovation is really at hand's reach. Being in departments offering good mentoring, support, and protected time is extremely valuable, especially in the early career where you can't finance your protected time alone so easily. Time abroad and out of the box is essential, in my opinion, very beneficial. My year in the US, my exchange with the lab of your shop and other labs was really very important. Collaborating with top researchers, especially if you're clinician scientist, you can't spend the whole day reading Science, Nature, JID, and you have to see patients too. You do want to be collaborating with a top group and profiting and interacting with them. That's definitely an advantage. Networking at meetings and being part of good research societies such as the ESDR is key. For me, it was a wonderful experience. Every ESDR meeting, also working on the board of the ESDR, extremely enriching. And then research in medicine is challenging and a wonderful experience. It has advantages being a clinician. Keep your eyes open when something is not clear, or when you see an effect of a therapy and it's not understood. Those can give you early hits for nice experiments that you can pursue in your lab. So I tend to see an opportunity in every difficulty. That's my mental nature, rather than see a difficulty in every opportunity. This is not a saying of mine. It's a wonderful saying of Winston Churchill that I really admire. And I think, yeah, in research, it can be tough, but it's good to look for an opportunity in every difficulty and just persevere. You will succeed down the line with perseverance. And with that, I come to the end of my talk, and thank you for your attention, and I'm open for any questions that may arise. Thanks so much.
Thank you very much, Lars, for this wonderful lecture. Just a quick question. I mean, you have mentioned this kind of variable results that we have got over the years with IVIG in toxic epidermal necrolysis, and I mean some of which has been attributed, as you mentioned, to some clinical variability. But do we have any molecular insights into what may also underlie this variability in the response to IVIG? Based on the data that we have reviewed?
Yeah, no, well, we do have molecular insights. We don't know if IVIG do bind to other cytotoxic molecules such as granulysin, perforin, or the activity of annexin on macrophages. So that is not known. But I think one of the big challenges, and if you look at some of the articles, the papers have been receiving a total dose of 0.4 grams per kilo. And when the data shows a linear relation between dose and protection against mortality with a significant effect as of 2 grams per kilo. So in Miami, who's been doing that, they have an intensive care unit. They've been doing it now for 20 years. They're treating with 4 grams per kilo and continue using that. So I think that's a problem. The literature is polluted with patients who have just received 1 day of treatment, patients who have been put on treatment when the skin is completely detached, and if you can't block apoptosis when the skin is completely detached, and some patients are getting low dose. And so I'm open, I may not be effective, although we have a pathomechanism behind that, but I think still we mustn't give up, and we must look at all these treatments in an open manner, and I hope the registry will contribute to doing that.
Okay, thank you. Maybe just one last question. Do we, I mean, you mentioned the fact that you have been studying clonality in the context of cutaneous T-cell lymphoma, but today clonality is becoming quite fashionable also in the context of inflammatory diseases. So how does that translate into clinical insights in terms of what can we take from those new data about clonality in inflammatory diseases to our patients?
Yeah, I think today the technologies that we have enable us to identify T cells with specific V betas that may play a role in a certain disease. And if we do identify these, well, technically down the line, we may be able to target them and delete one single clonal part of a response that may be pathogenic in disease. That technology is being developed down the line. But first, we have to clearly show that one V beta is driving one type of disease. It may be complicated because we're also learning in inflammatory disease very clearly, and that through transcriptomics analysis, that diseases are really heterogeneous, and even diseases like psoriasis, and even more the case, more typically in hidradenitis suppurativa, if you do transcriptome analysis, you see there are subtypes, or so-called endotypes of disease, with slightly different signaling pathways, and this probably explains why not everyone with an HS responds to anti-TNF. And I think that's something we have to look into more detail. And I could also imagine that in diseases where one V beta does have a pathogenic role, it might not be the same V beta in another patient suffering from the same disease. So this is a really interesting field of research that should be looked at with the technology that we do have today.
I'd like to very much thank you, Lars, for this fascinating session. And I would like to invite you all to the next ESDR Kitchen episode, which is due June 16. It will be part of our Freshly Baked series. Mary Tulik and Emma Chambers will update us on novel aspects of the skin microbiome and human skin aging. So please do not miss it. Thank you all for having been with us. Wherever you may be, please note that the abstract deadline for our next ESDR virtual meeting in September has been extended to the 5th of June. So please explore the opportunity. Goodbye, everybody, and thank you for attending.
Thank you very much. Bye-bye.
Bye-bye.